And it's now my great honor to introduce our keynote speaker, Dr. Steve Holland from the NIH. Steve, going back in his history, went to college at St. John College, one of the great books curriculum in Annapolis, Maryland. And then he went on to medical school at Johns Hopkins University, where he stayed for his internal medicine residency and became a chief resident. After that, he did an infectious disease fellowship and went on to the NIH, where he was exploring aspects of HIV infection. Got interested in phagocytic biology and phagocyte immunodeficiencies. Since 2004, he's been chief of the Lab of Clinical Infectious Disease at the NIAID. In 2011, he became Deputy Director of Intramural Clinical Research at the NIH. I've known him for years for his work on mycobacterial infections, many talks I've heard on chronic granulomatous disease, identifying new pathogens found in that disorder. More recently, he's been working on hyper-IgE syndrome, and today we'll hear about a new clinical immune deficiency disorder that's been figured out just in the last few years. Thank you.
Okay, you know, before I get started, I'd just like to thank, well, I'll find the right slides. If not, that last talk was so good, I'll give it again. I mean, you know, I'd like to just thank the organizers for inviting me. And I'd also like to say that this is the first Immuno-Hematology Comprehensive Program day. And my God, if this is what they do on their first day out, what in the world do you think they're going to be doing a couple of years from now? This has been extraordinary. And so I'd like to give, you know, a special shout out to the director of the program and to, obviously, it's the importance of grabbing together and collecting a team of people who are all committed to the same principles that really makes for success. And I just could not be more impressed. So it was my great pleasure to meet Kelly when she came and spent a few months at the NIH. And I really was tremendously impressed with being able to work with her and to have a hematologist who was interested in immunodeficiency. And I know people in immunodeficiency tolerate having infectious disease people involved, so I know how important that is. But I also know the real reason that today was chosen for this symposium, and I know what's coming. Okay? Now, a lot of you know just how kind and gentle and thoughtful hockey players really are. This is, of course, the game for tonight. You got the good guys and the other guys here. But, you know, the important point is that they're really gentle people. And you see, I mean, here they are exchanging tender words because there's nothing that makes for allies like common enemies. And you know, some guys waste their time shooting the puck, and other guys know where the real action is. All right, so it's really a great honor and a pleasure to be here to talk about a disease that I think is really exciting. And in part, it's exciting because I know that you've been seeing it. And I know that it's a disease that somehow has, over many years, because it's presented in so many complicated ways, been missed. And I think we've heard example after example of that today. The XAIP story is an absolutely fascinating one. I mean, here you have people that show up with everything from colitis to HLH. How do you put that together? And frankly, we put it together by recognizing genes. And what I want to talk to you today about is a genetic syndrome that we've put together after years of looking at it. And just as was pointed out by Colin earlier, this disease has been recognized for a long time, but recognized in different ways. So I need to remind you that phenotype is how we all work. Phenotype is what we see and how we collect our thoughts. But phenotype can be misleading. And so you know what you're going to call those little penguins when they figure out that the phenotype is not the same as the genotype. That gets called lunch, right? Because sometimes what you see on the outside is not what counts on the inside. So I want to take you on a brief tour of what it has been to come to understand this disease. And this is just a picture from Midsummer here in Ann Arbor. And I want to just sort of walk you through these things from the start. So the story I want to tell you starts in 1984 in Boston. And it starts with the recognition of a girl, a child, who showed up with severe, overwhelming varicella. And she showed up with her primary chickenpox, covered in lesions, with varicella pneumonia. And on taking her history, it was clear that she'd not really been completely normal all her life. She'd had recurrent otitis, well, I mean, come on. And she had had other problems, and someone had noticed over a few years that she had leukopenia. But no one had done anything about it because she was otherwise doing okay until she got the primary chickenpox. And following that, she did not do as well. And over the next few years, while people were sort of gearing up to the idea that she was going to need a bone marrow transplant, she died. But this was 30 years ago. We weren't as good at doing those things back then. And the group of John Sullivan, working with Christine Byron, who was then a graduate student in the laboratory, recognized in retrospect that what made this girl so remarkable was that she had no NK cells. Now, you just heard a spectacular talk about just how important NK cells are. And this was really the first human experience saying, oh my God, those cells are important in handling things. And they realized that having no NK cells in this girl was probably linked to her viral susceptibility. And 5 years after the girl showed up, they published this paper here in the New England Journal. And what they showed at the time was that she had impaired ability to lyse NK targets. So I told you that she had no NK cells. Well, of course, no is a relative term. She had a few NK cells, and if you purified blood aggressively enough, you could get some cells. And then what you could show is that her ability to lyse targets, here we're looking at either K562 or Dowdy cells, but she's unable to lyse the targets, whereas the normals are doing just fine. And it didn't matter what you did. She could not quite get it right. That was 1984, and then up to 1989. And right after that, Stu Orkin, working with Doug Engel and others, started to look at chickens. Now, of course, you might say, you know, why jump from humans to chickens? Because that's where the money is. And they realized that there were 3 proteins, all of which looked virtually the same as transcription factors, all of which were being expressed in the same cell. And they said, wow, that's weird. And they posed the question, why are there 3 different transacting factors recognizing the same consensus motif all within a single cell? That's a really, really good question. It sets up a lot of competition, all within the same cell, just like many PIs do when they have a lot of graduate students in the lab. And we all know how happy that makes people. So the following years, they recognized that these transcription factors were binding to a site shown here. Now, these are assays that nobody does anymore, so I won't even try to explain them because they're irrelevant now. But, you know, they recognized by nuclease protection that these factors were binding to a specific target sequence, GATA, hence the name GATA. And they finally decided that, as I mentioned, there were 3 of them inside these cells, and they called this particular one GATA2. And what was really important about GATA2 was that it turned out you had to have GATA2 in order to induce the proliferation of endothelial cells. And so this is the protein that regulates pre-pro-endothelin 1 in endothelial cells. So without this, you get no production of endothelium. Well, Stu Orkin, who has just been a demigod in this field, went from the recognition of the importance of vascular regulation to recognizing that the cells in which this was being produced were, in fact, hematopoietic stem cells, and recognizing that GATA2 was critical, along with GATA1, for the development of erythropoiesis. And so he spent the last 20 years looking at erythropoietic functions of the GATA factors. And, you know, frankly, painful to me, ignoring these little guys down here, these here things that we call white cells. Now, it is a great privilege to be among people who think that those are important. So that was 1990 and 92. So in the same year, I want to move down to Bethesda, because in that year, we started seeing patients at the NIH who had widely disseminated non-tuberculous mycobacterial disease. And this is the 2 patients from the first paper that we published on this. And what's important here is that we had 2 people, here a boy and a man. This boy was actually the first case referred to me. I had moved over to the NIH to work with John Gallen, who was a CGD expert. And John said, Steve, you come into my lab, you'll have a great time. All you have to do is, number 1, take care of the patients that I take care of. They've got these funny diseases called CGD and Job syndrome. He says, that's easy. And then, you know, if you want to succeed here, all you got to do, it's real simple, just go find your own disease. I said, really? They're just out there to pick up? So as I was working, I got a phone call from a guy in Florida, pediatrician driving a convertible with a cell phone, which was a big deal in the early 90s. And he says, hey, I got a family with CGD. I said, okay, we do that, okay. He said, you know, it's a funny case, though, because the kid's got disseminated mycobacterial disease, and he's got 2 maternal uncles who have the same thing. You want him? I said, well, sure, of course. You know, I'm a government employee. Of course I want him, yeah. But, you know, that didn't sound like chronic granulomatous disease to me. But I said, well, that's fine. You go ahead and send him up. Well, it turned out that this kid had widely disseminated mycobacterial disease and interferon gamma, which had become very interesting around the time because it had just been approved for the use in chronic granulomatous disease. We knew that it would activate macrophages, so we gave him interferon. This kid had really impressive mycobacteremia. Here we started interferon. Suddenly he started to grow, which he hadn't done for years. And he'd been on TPN because he had such severe diarrhea. And we cured his diarrhea. We made him grow. We cured his mycobacterial disease. I mean, for me as an internist, you know, internists, we don't cure much of anything, right? So this was a big deal. And we started collecting patients who had disseminated mycobacterial disease. And we got this guy who came in, and this is the taps of his chylus ascites. And this is every week, right? So here is these taps here for 6, 5, and 4 liters of ascites every week. And we start him on interferon, boom, his ascites goes away, and he's cured of his mycobacterial disease. Well, you can imagine that what that makes you do is say, give me more. Let's find out who's got what. And of course, what then you find is that there are those people who follow the rules that you set up and those people who don't. And so as we recognized this group of people who responded to interferon, we found that there were 2 patients in our initial cohort from the early 1990s who were funny because they had very few monocytes and very few B cells, even though they had histiocytes and plasma cells. And we were really puzzled about that. And so, like so many things, we started out with some things that we thought we understood, and suddenly you run across something you don't understand. And then you have to make a choice. Do I follow what is unsure and uncertain, or do I go with what I know? So here's patient number 5, the 5th patient that we recruited who had severe disseminated mycobacterial disease. And what you see is that she was in her 40s. She had this very large lesion here that's just chock full of mycobacteria, and she had these lesions all over her body. And this pus was easily detectable to have mycobacteria. And she complained of a little headache. And you can see here in the back of her left orbit, she had a tumor. And when we resected that, it was a smooth muscle tumor that was chock full of Epstein-Barr virus. You're not supposed to have Epstein-Barr virus outside of your, upper pharynx epithelial cells or B cells. That is fundamentally immunodeficient. Her course was really quite unusual. She had had pneumonia in her 30s for which she had had a lobectomy. Now, I'm not telling you that lobectomy is how we treat all pneumonias, okay? It's not even in my internal medicine practice. But it had happened because she really had refractory problems. And then she developed disseminated mycobacterial disease. And when she came to us, we recognized that she had no monocytes, no B cells, no NK cells. Despite that, she had normal immunoglobulin levels, and her T cell number and function seemed to be okay. She had had severe perineal warts, and when we met her with her disseminated disease, we treated her with interferon gamma, and like the other patients, she cleared her disease. In the process, though, she developed chronic myelomonocytic leukemia, and when we referred her to bone marrow transplant, again now 20 years ago, much less effective than it is now, she developed a community respiratory virus and died. But one of the things that we really focus on at the NIH, and I don't know how you do it here at Michigan, but the importance of autopsy cannot be overstated. If we don't learn from our failures, it's extremely hard to learn from our successes. And so we're very lucky to have the ability to bring patients who die elsewhere back for autopsy and to try and talk to them sometimes ahead of time, saying, you got a bad problem. You may not make it, but this is your investment in the future. Autopsy rates are going down in the United States because everybody's sure they know what's going on. But I live around a lot of people who are sure they know what's going on. They're called congressmen. And I can tell you that injecting data into that is never a bad thing. So after our patient died and came back, and we found that she had in her tissues, as I mentioned, here is macrophages, even though there were no monocytes in the peripheral blood, and here are plasma cells, even though there were no B cells in the peripheral blood, and immunoglobulin levels were normal. Going back to her family history, here is our patient, and it was striking that her mother had died in her 50s of chronic myelomonocytic leukemia with disseminated mycobacterial disease. In the process of working her up for bone marrow transplantation, we examined all of her sisters, and we found that this sister also had low monocytes and low B cells and low NK cells. So we didn't think that she'd be a good donor for transplant, even though we didn't know why. And interestingly enough, in her late teens, she had developed severe perineal warts and had had to have several laser procedures for that. Otherwise, she felt fine. About 3 years later, this sister here, who had survived and was now in her mid-30s, came back because she too had developed fever and disseminated mycobacterial disease. And we treated that, we cured her disease without having to use interferon because we caught it very early. And in the process of working her up, we realized that her chest CT wasn't quite normal. And you see here that there are some small nodules that might be referred to as tree in bud. That is, things that look like there might be active processes going on out in the periphery. We offered her the opportunity for an open lung biopsy. She said, "Are you crazy?" She felt fine. She was breathing fine. She lived in Denver, had no problem with doing all of her activities. We said, not a bad decision. Go about your way. But a few years later, she came back. And now, her lung disease was not so subtle, and her oxygenation was not so good. But what she had was not active infection. It was pulmonary alveolar proteinosis. And what you want to remember is that pulmonary alveolar proteinosis occurs when pulmonary alveolar macrophages fail to digest surfactant. And so this occurs, the most common cause in the United States now is autoantibodies to GM-CSF, which she did not have. Now, despite the fact that she didn't have those antibodies, she did have a lot of goo in her alveoli, and we had to wash that out. And so we did what is standard practice, which is a whole lung lavage. And here, you know, what you need to do is to get a hold of a pulmonologist who has nothing to do for a full day. And they have to go in, they have to intubate selectively, and then wash through here 20 liters, starting here, ending here, going from, you know, latte to whatever, and wash out all of the surfactant in the lung. And when we looked at that in the laboratory, what we could see was that indeed here too, despite the fact she had no monocytes in the peripheral blood, she had plenty of alveolar macrophages, each one of which was chock full of surfactant and things that she should have been digesting and excreting, which would have kept her lungs intact. So we didn't know what to make of this, and after studying this disease for a long time and coming up with no particularly good answer, we decided we'd better describe it phenotypically so that we could organize our own thoughts about it and try and, you know, see if other people had encountered the same thing. And so we referred to this as monomac, monocytopenia with mycobacterial disease, and characterized by being missing these cells, having these infections, so MAC, but also histoplasma and viruses. Some had aplastic anemia. Many of them went on to develop myelodysplasia with leukemia, and then pulmonary alveolar proteinosis, and in the setting of PAP, some very severe pulmonary hypertension. So let me just show you what some of this looks like. This woman has, she's in her 20s, otherwise very healthy and functional, but has very severe warts over her hands and in her perineum. This man is 26 and has here this lesion in his liver, which on biopsy we thought it was going to be an abscess, but in fact was a smooth muscle tumor that is diffusely full of Epstein-Barr virus. This man had severe histoplasma infection, had a big abscess in his psoas muscle, and you can see this is all histo. They never get pneumocystis, even though it looks like that. And unfortunately, this man went on to get another fungal infection, this one here called Neosartoria uragawa, which is a very rare but highly invasive fungal infection. His mother, the same man, his mother in her 60s was completely asymptomatic except for having unilateral lymphedema with these diffuse flat warts on her legs. So how to put all that together? Well, you know, we didn't know how to put it together, so what we did was just describe it. And describe this as this one syndrome characterized by these things that I've mentioned to you, including a fair number who got erythema nodosum, and then a very substantial number who died over the years that we'd been following them. So we knew we had a bad disease, we knew that it was involving many different pathways, and we knew that we didn't know what was going on. So one of the most important things about working in an institution, what makes it fun, what makes it important, is that you run across people who tell you things you didn't know. And of course there are a lot of things that most of us don't know and we need to go to experts for. And so I was very lucky to work with Kathy Calvo, who is an immunohematologist at the NIH, who said, you know, these guys' marrows aren't normal. I said, well, yeah, thanks. You know, I sort of knew that, thanks. But she said, no, 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 they're not normal in ways that you didn't know about. And so she said, look, let me show you here on the flow cytometer. So this is what our normal marrows look like. Here's the granulocyte gate, and you can see all the granularity in the neutrophils. Here are the monocytes, lymphocytes, and blasts. And what she said is, look, your patients are really abnormal because, yes, they have granulocytes, but they're very hypogranular. That suggests that the defect affecting their myeloid series must actually be occurring early in myeloid ontogeny, because the granules get made very early in neutrophil development. And sure enough, not only do they not have monocytes in the peripheral blood, they don't have them in the marrow. But wait a minute, I should have been stopping and saying to myself, wait, how can that be? Because they got them in the tissue. And up until this point, we had been thinking, how can they be not in the blood, but in the tissue? We thought at first, maybe they were being sort of sucked out into the tissue. Maybe they were over-avid for going out into the tissue. And as we started to do flow on the marrow, we said, uh-oh, wait a minute. That's not it. They're not making them. But they must have made them once. And then she said, you know, the defects are not limited to that. It's not just in the myeloid series. Look what happens in the lymphoid series. So here we're looking at normal B-cell development. And to make a long story short, normal B-cell development should start with CD10 positive cells and end in CD20 positive B cells. And these are referred to as the hematogones. And what was abundantly apparent in virtually all of our patients was that their immature B cells were gone, and all that was left behind was a few mature B cells, as if everything fertile had disappeared, leaving only the old folks like us around. So this was really a puzzle. When we go back to look at the marrow itself, what you see is that it is characteristically hypoplastic, and then you see that they've got these atypical megakaryocytes. Normally, megakaryocytes should have nuclei that are close together because of endoreduplication. In fact, these have megakaryocytic nuclei that are widely separated, and in fact, some of them are abnormally small, giving rise to the genuinely oxymoronic term micromegakaryocytes. So, now we got to go back to our patients again, because I don't know how to do things that aren't patient-oriented. I'm very simplistic in that way. And so we go back to our patient, and we can lump together her and her sister. We're talking about problems that include HSV and EBV and pulmonary alveolar proteinosis, disseminated mycobacterial disease, disseminated warts, as well as leukemia. How are you going to put all that together? How are we going to put together all of these different features, including lymphedema, in an autosomal dominant pattern? So that means, right, it's got to be one gene. And if it's one gene, there are only 23,000 to choose from. Sooner or later, we got to be able to find it. So, for those of you who either have read the book, The Berenstain Bears, to your children, or are young enough to have had it read to you, there's that one wonderful book where the papa bear constantly tries to tell the baby bear how to do things right, and each time he gets it wrong. He tries to ride the bike and he crashes. He tries to fix things and they break. And he ends each little section saying, "Baby bear, don't do it this way." So I spent 20 years finding all the wrong ways to look for this gene. And finally, the technician in my laboratory, Amy Sue, who's extremely gifted, said, "You know, you are being very thick-headed about this." I said, "Well, again, thank you. This is really very supportive." She said, "No, look, you have to rethink this because your patients have problems in erythrocytes, megakaryocytes, granulocytes, monocytes, dendritic cells, B cells, and NK cells. There's no way that can happen anywhere but at the stem cell level. You've got to stop looking down here, which is what, I'm just an ID guy, you know. We stand around, we talk about what's the absolute count of X or Y or Z. She said, don't do that. You got to start thinking the only way to catch all of these things in the same net is to go up to a hematopoietic stem cell. And she was absolutely right. And so we started sequencing through those, and on May 3rd of 2011, she called me up at 7 o'clock in the morning and said, started screaming into the phone, 8 out of 8, 8 out of 8. I thought she'd like hit the lottery. I didn't know what it was. And I said, "Excuse me." She said, "All 8 of the patients we sequenced have a mutation in GATA2. Well, okay, so what is that? How does that help you? So remember, we started talking about a factor that binds to the GATA sequence. So that's a 4-base sequence in the genome. Hold on. This is where it gets really exciting. So don't worry, that's all. But it's really, I mean, so it's a 4-base sequence in the genome. There are 1.7 million gatocytes in the human genome. 1.7 million. So it's hardly a surprise that this thing is important. And so, you know, here we have GATA2 right at the center of the universe, of course, and what it's controlling, and we'll go through these in the next couple hours, but what it's controlling are all these other factors. So it's a transcription factor. And in retrospect, of course it's a transcription factor. How can you get this complicated, things that are somatic and immune and developmental, without controlling transcription at an early level? And in fact, this controls other transcription factors, histone deacetylases, endothelial nitric oxide synthase, as well as angiopoietic factors, and many other factors in between. Well, we were very lucky at the time that when we saw this in our patients, I had been working on this since 1992, and so it only took me from 1992 to 2011, just 19 years, to get out a good paper on this subject. Now, I am not suggesting this is a model for graduate students and postdocs to follow, but you know, it is important to be able to have some of the freedom that's required to accumulate things over a long time, because not every result comes up quickly. Well, as is almost always the case, you know, good ideas are rarely held alone. And so at the same time that we recognized this in our syndrome that we called monomac, well, you know, there was just a flurry of other people with exactly the same findings. So here, Marshall Horowitz and Hamish Scott had been looking at families in North America and in Australia. This is a 4-generation family in North America, and they said, boy, these guys get leukemia that comes on out of the blue and is familial. Now, I mean no offense to geneticists, Mark, or oncologists, like everybody, even my own dad. But I gotta say, when oncologists say that cancer comes on out of the blue, sometimes that means they didn't ask about anything else that wasn't cancer. You know, I know that nobody here is myopic, but you know, where I come from, people focus on what they focus on. And sometimes you got to go back and say, was there anything going on that I didn't ask about? So in this family, things like warts and mycobacterial disease sort of got missed. It's not like it was a different disease. But the group of Matthew Collin in Newcastle was asking the very different question. They said, what do people that have no dendritic cells look like? And as they trolled through their populations of patients, they found 4 people who had no dendritic cells. And sure enough, they had exactly the same disease that we had. They came up with the name dendritic cell monocyte and NK lymphoid deficiency, DCML. I would submit monomac was a better name. Okay, but anyway, that was their name. And they said, well, gee, look at that. They got no dendritic cells in the peripheral blood, but they do have cells in the tissue. And then the group of Sahar Mansour found the same gene in a completely different disease called Emberger syndrome, characterized by lymphedema and myelodysplasia and leukemia. At least in this particular case, we've got something of a mechanism because we know from mouse work that GATA2 is being expressed right here in the lymphoid valves, the valves of lymphatics, suggesting that GATA2 dysfunction in lymphatic valves might be involved with lymphedema. Finally, I was sitting in a talk from somebody from Jordan Orange's lab, and she was going back through all the different kinds of NK cell deficiency and presented this case again. And I sat there and I listened and I said, wait a minute, you're talking about somebody who's got low leukocytes and bad varicella zoster and then went on to die? I'll bet this is GATA2 deficiency. And in fact, I started calling around to everybody I could think of, and I called Jordan Orange, he didn't have any cells, and I called Christine Byron, she didn't have any cells, and I finally got to John Sullivan. He said, oh yeah, I got 1 vial left. Now, he might say that to everybody, I don't know. But he had the 1 vial that he sent me, and sure enough, this same girl that had been reported 20 years before had, in fact, a mutation in this same gene. So now we're in this funny place where here it is, the summer of 2011, and suddenly we've gone from we've got no genes to explain this to, you know, a pile of different syndromes that all look like they're targeting the same gene. So how can one entity have so many manifestations or interpretations. That is, how could everybody be reading the same playbook and come up with such a different explanation for it? And I would just point out that this is happening all over the world for a long time. So it's not like we came up with confusion ourselves. But I think what we ought to be doing is referring to this as GATA2 deficiency, not by any of these other phenotypic names, because this really explains all of them. No matter which name you started out with, this is it. It's 5 names, but 1 disease. So let me go back now and talk to you for just a minute about GATA2 and what that does. So GATA2 is your typical 2-finger zinc transcription factor, you know, fingers 1 and 2, and it straddles the DNA and recruits in other transcription factors. And what makes GATA2 the GATA factors so interesting is that there are 6 of them in humans. And they fall into 2 big groups, the hematopoietic ones and the more somatic ones. And GATA1 is something that shows up in Down syndrome with anemia and thrombocytopenia and a myeloproliferative syndrome. GATA2 we're talking about. GATA3 virtually exclusively stays among endocrinologists and nephrologists, where it often shows up as hypoparathyroidism, deafness, and then eventually renal disease. These factors are much more involved in cardiac formation and other features. Now, as I mentioned, there are 1.7 million GATA binding sites in the human genome. And to make it even more complicated, these are relatively promiscuous factors. Let me show you why. So here we've lined up all 6 of the GATA factors. And in red is every place where there is amino acid identity. So what this shows you is that the zinc fingers are virtually the same among all 6 of the GATA factors. But there's a lot of diversity as you get out here into the C and N termini, which are recruiting distinct and different transcription factors. Now, just to make it even more complicated, the GATA factors, Gesundheit, don't act where everybody else acts. So as I grew up thinking about genes and genetics, I was much more interested in exons and promoters, and I was sure that that explained like 99% of all the world. But in fact, in elegant work by Emery Bresnik, who has been just a world leader in this subject, in fact, what turns out is that GATA factors really don't care much about exons, and they really don't care much about promoters. They spend most of their time binding in introns and in enhancers, precisely the areas that you never collect when you do whole exome sequencing. So now we've got an answer, GATA2, and we've got a transmission pattern, autosomal dominant, and we know it's heterozygous. So now we've got to think for a minute about what's really going on. So you get 3 choices for how a heterozygous dominant mutation could work. It could be a dominant negative, inhibiting the function of the normal allele. It could be a dominant gain of function, conferring a change that was not anticipated. Or it could be haploinsufficient. It could need to have both alleles present. And I'll spare you the painful work of going through this, except to point out that this disease has missense, nonsense, and intronic mutations, all of which look virtually the same. So just the one really important one here is in the intron. That is, GATA2 binds to itself and induces its own expression. And so there's a discrete set of mutations here in the intron that eliminate the ability to enhance GATA2 expression. And so from that allele, even though the message is perfectly intact and splicing is preserved, the level of expression is profoundly diminished. So when you see all of these things, missense, nonsense, and intronic mutations all together, that means that the mechanism behind this is haploinsufficiency. That is, one allele just is not enough to get the job done. And we can show this at a functional level using a nanostring assay, that the level of transcript, no matter how you mess up the transcriptional program, either with missense, nonsense, or intronic, you wind up with virtually the same thing, which is too few copies. So haploinsufficiency turns out to be the mechanism by which it works. I just point out that the work that Joanne presented earlier suggests that there is an element of haploinsufficiency for Artemis as well, which is why you have a heterozygous phenotype that confers lymphoma, whereas the homozygous phenotype confers SCID. So what are the consequences and manifestations of this? Let me just run through these very briefly, and I promise to get you out long before last call. So viral infections are very common in this disease, and they start in childhood. So somewhere in late childhood, either before age 10 or sometimes in early adolescence, children develop bad warts. And these aren't just your average warts that come and go. These can be warts that are usually digital, but occasionally can be perineal, and they can be disseminated otherwise. They're not as profound as in some of the conditions like DOC8 deficiency, but they're really bad, and they annoy them. And you can see that over time, it starts around here, and then over time, virtually everybody develops warts. And here is just the different colors reflect the different kinds of mutations, trying to show that there's not very much variation between whether you've got a missense or a nonsense or a null mutation. And following the wart infections a little bit later in life, starting later in adolescence, they begin to get fungal and mycobacterial infections, and you see these accumulate over time. And then following that, later on in life, come the myelodysplastic changes. And these are really quite difficult, and that's when, I mean, they certainly come to attention when they get mycobacterial disease, but they really come to attention when they get myelodysplasia. And after the myelodysplasia, usually a few years afterward, that's when the pulmonary alveolar proteinosis begins, presumably because this is when those macrophages that got made in the marrow as monocytes in childhood went out and populated the lung, and then were just abandoned there. Like all of those movies of, you know, the astronauts who wind up going to a planet and then the Earth explodes and they're left there talking to, I don't know, their cell phone or, you know, to somebody else. And those abandoned macrophages can work for a while, but not forever. And it takes a long time for them to become effete and incapable. And then the other thing that we've learned by studying this disease is that there are many different features, many different things that GATA2 is regulating. So one of the most important, working with Jordan Orange and Emily Mace at Baylor, is that you really need GATA2 to develop the CD56 bright, highly cytokine-producing population of NK cells. And we can show that even if you start with CD34s and drive those to NK cells, they cannot make the bright population. So these patients are, when they have their NK cells, they only become the NK dim ones. And then over time, they lose even those. All right, so what other manifestations here? The hypoplastic myelodysplasias, I would emphasize that these are likely to be where you're going to find this disease. And they can show up as MDS, as aplastic anemia. 10% of the cases of neutropenia in the French registry turned out to be GATA2 mutated. And then we've talked about monocytopenia and B or NK cell cytopenias. Just because it wasn't complicated enough, let me remind you that it can have very variable presentation. So the penetrance of this disease is complete. Everybody who gets the gene gets something, but not everybody gets the same thing. So penetrance is complete, but expression is quite different. So in this family here, all 3 children developed myelodysplasia. All 3 developed trisomy 8. 2 of them had severe varicella zoster. 1 had severe HPV. 2 of them had DVTs in childhood. And by 18, all 3 had been transplanted. So you knew there had to be a parent who was affected. And yet their 55-year-old father is absolutely healthy, feels fine, no problems, and yet his marrow is not so normal. He's got the micromegakaryocytes, he's got 2-cellular marrow, and yet he's doing fine. So you never would have picked him up on a straight phenotypic screen or a symptom screen, whereas these kids were obviously in big trouble already. In addition, there are these changes here. This boy came to us. He was an Afro-Caribbean child who came up for severe aplastic anemia, was treated with standard immune suppression. 2 years later, he comes back now with MDS and monosomy 7 and rapidly evolved into AML and died. And he has a typical GATA2 mutation. As we start to collect these patients and start to try to understand this, 2 papers were presented at ASH in December, one from Alison Bertucci at Baylor, looking at sporadic, non-syndromic pediatric MDS, showing that the majority of those were GATA2 mutated, and then from Ingo Hoffman at the Boston Children's Hospital, looking at pediatric MDS, showing that a substantial number of those were GATA2 mutated as well. This is a big deal because when you're starting to decide who should be the donor for your child that has AML or MDS and needs a transplant, keep in mind that you may not know about the sibling. And I know of at least one case where a twin was used and the recipient died 6 months later, and 15 years later, the donor was in my clinic with disseminated mycobacterial disease. So doing this genetic testing is not so trivial. There are probably other presentations. This report of hypoplastic MDS with B-cell leukemia in early childhood from some children at St. Jude's. We don't have the genetic testing here, but I think in retrospect, these must be patients with this disease as well. Okay, so we're almost done. You've survived, and I promise we're almost through. So let me just say that even as an internist, no matter what you think of us, it's not gratifying to go into patients and say, wow, I really know why you're sick, and now I can tell you why you're going to die. That is not a winner. You cannot just work on cause. You have to work on effect. And so it's critically important to be allied with people who can help you to make things better. And so working with Dennis Hickstein and Jennifer Cuellar at the NIH, we've been able to really begin to expand a transplant program. And what I'm trying to show you here is that these people go in with no NK cells and get normal, no monocytes and get normal very rapidly after transplant, indicating that the problem is strictly in the hematopoietic stem cell, not in the stroma. Their soil is fine. It's the seed that's a problem. And when you correct the marrow, you correct a lot else. This was our first patient who had severe PAP going into it. This is after he had been lavaged, and he was on 6 liters of oxygen and high-dose sildenafil. After transplant, 6 months later, he's on room air, off sildenafil. His pulmonary pressures were normal. So repopulating his lung with effective macrophages was enough to get him better. Now finally, as we tried to figure out why are people getting sick, who gets sick and who doesn't, we realized that there were people that show up, they show up to the doctor because they're sick. But once you get a gene, you start to screen families and you start to find people before they're sick. And now you can ask a very different question. And so what we did was to go back and with Michael Spinner, who was the star medical student who rotated here with Kelly, we simply made a very simple score and said, if you have no problems, you get a score of 0. You got 1 problem, 2 problems, 3 problems, 4 problems, just problems around GATA2 deficiency. And what was interesting was that when you had no problems, you had normal numbers of B cells, NK cells, monocytes, or CD4 cells. But as you accumulated problems, your counts went down. But that puts you in the funny position of trying to figure out what is the chicken and what's the egg. Is it the cytopenia that makes them susceptible to infection, or the infection that makes them get cytopenias? Well, there's no way to do this without talking about mouse models. And so what you have to do is to then say, okay, what do we know about this? So Colin showed you a very interesting slide before about serial transplantation in some of these immune dysfunctions. And what we're looking at here is really the functional ability of stem cells in mice that are heterozygous or not, heterozygous or wild type for GATA2 deficiency. And what you see here is the ability of wild-type animals to do serial transplantation, whereas animals that are heterozygous for GATA2 mutation are very poor at transplantation. When you look at the secondary transplants, you can see wild-type go down a bit, and the heterozygotes are in big trouble. Animals that are knockouts for GATA2 have no ability to form a marrow at all. Well, that doesn't really answer our question. We have to go to volunteer studies, and recruiting volunteers is sometimes difficult. And the consent, reading the consents to these guys is very tough. But what we decided to do was to try and induce the same kind of disease in mice that we were encountering in humans. And so we gave them intravenous mycobacterial infection in the tail vein and then sampled marrow over a period of time. And I'll summarize that in just one slide here and point out that after 16 weeks, what we could see is that our heterozygous mice had very few stem cells left, whereas the wild-type mice had plenty. That is, and when we didn't infect them, their marrows were fine. So this suggests that there's at least an important element of marrow consumption driven by infection, a marrow exhaustion. All right, well, I've gone on long enough. I'll just point out that it's not a simple story, because there are some cases in which GATA2 mutations are actually a good thing. So if you've got a CBP alpha mediated leukemia, having GATA2 mutations in that context actually is protective. Those children do better. In contrast, if you're an adult and you've got a GATA2 overexpressing leukemia, you do worse. So the roles of GATA2 are going to be different depending on where you start and what you got and how things go. So, you know, I like to think of this as the typical story of the blind men with the elephant, or maybe women too, although women tend to be more insightful than this. But, you know, it looks like many different things to many different people depending on where you come from. It can be myelodysplasia, pulmonary alveolar proteinosis, mycobacterial disease, warts. People start out normal, lymphedema, fungal infection, or acute leukemia. All of them the same genetic problem. So how does GATA2 do all this? Well, it's got lots of effects in lots of tissues, abnormal vessels and lymphatics, overlapping targets, overlap probably between GATAs 1, 2, 3, 4, 5, and 6, early population of tissues with macrophages and plasma cells, and then the immature cells die out and people are left without them. They have impaired definitive hematopoiesis, infection-driven bone marrow exhaustion, and then there are lots of secondary mutations and cytogenetic effects. So GATA2 deficiency, it's more common than I thought and more common than I think we still appreciate. It's got multiple manifestations that cut across disciplines. There is a lot of infection that goes with it, but it can start out as any of the ways it wants. It's got a variable onset, but the cytopenias tend to be progressive. It's one of the really outstanding examples of haploinsufficiency with regulatory mutations and clear evidence that we are focused on tight control of this gene. It's taught us a lot already about NK cell deficiency and dysfunction, and we're now looking to try and understand the rest of myeloid function through it. So here's one gene that ties together infection, hematopoiesis, immunodeficiency, lymphedema, alveolar proteinosis, and cancer. And there just aren't that many genes that cut across that many disciplines all at once. So in the last slide, I need to just remind you that genotype is a unifying feature, whereas phenotype can change over time. And there are many things that can make this happen. You know, time, age, exposure, even satisfaction can lead to differences in how these things manifest. So lots of people contributed to this work, and they really have been outstanding in it. In particular, Amy Sue and Elizabeth Sampaio in my lab, Lauren Sanchez and Michael Spinner. Without them, we really would not have gotten all the phenotyping done. And especially my collaborators, Emily Mace and Jordan Orange and Jennifer Cuellar Rodriguez, and Dennis Hickstein. Thank you very much.
We have time for a few questions. Steve?
What is it that's compensating for mutations early on in embryogenesis and early childhood?
If I could rephrase your question, why is it that they start out normal and then end up abnormal? And boy, I really wish I knew that. And I have to think that there's some, I mean, when you look at the level of stem cells, when you look in the mice, the level of stem cells in the mice is 50% in the heterozygotes. You know, I mean, really, the normals are here, the heterozygotes are here. Despite that, a significant number of people go on for a long time with no trouble. And maybe it is my bias as an infectious disease-oriented person and my bias because the way they came to me was all through infection. But I tend to think that their marrows are precariously compensated. They're able to reproduce at a certain level. And when you stress that with things like certain infections, you really get into trouble. Now, the proof of that is really going to be through mouse experiments. And I have a gifted young oncologist in the lab now who's trying to do this by looking at chemotherapeutic challenges and then crossing them onto backgrounds that have you know, other components as well that might help us understand just what is it that drives the myelodysplasia. Matthew Collins' group that started out with the question, what do people look like who have no dendritic cells, recognized early on that FLT3 ligand levels are high in these patients, and that as their myelodysplasia increases, their FLT3 ligand levels go up. So I don't know, I mean, maybe that means that FLT3 is pushing harder and harder and harder on that marrow, and it can go just so far before it runs out of gas and exhausts. I'm much more clear about what's not a problem here than what is, and telomere length, which we were very interested in early on, turns out to be normal, and I think not so affected by this defect. But your question is an absolutely central one that I don't have a good answer to yet. Sir?
In light of that, isn't it, I mean, there's a lot of leukemias that occur in our patients early on that we don't see in adults, and vice versa, things that we don't see, and so maybe it's just the dependence that GATA2 is not important early on in development, and is important later on in development, in adults, sort of developmental sort of things. And you see that, the leukemia that right after birth are very dependent upon what they got from the mother. A little bit later on, they're not as much so. By the time you get adults, it's a lot less dependent on what you sort of had there originally. So maybe it's just a developmental program that, the GATA2 is for some reason more important later on in life and not as important early. Is that not a way to think about it?
Well, first off, I could never say that GATA2 is not important. I mean, just, flat out. But I think that it's going to be one interesting set of experiments. If you knock out GATA2 entirely, okay, so you make a homozygous knockout, those embryos die at about 6.5 days with no hematopoiesis. If instead you take those intronic mutants that are hypomorphs, they express about 20% of the normal allele, you make a homozygote with the hypomorph, they live until 13.5 days, and they make a marrow, but they don't make the vessels to pump it around. So I think GATA2 is critically important at many different levels, but having a 50% expression gets you past certain milestones. Now, you're certainly correct that I don't think this is showing up as those, early onset pediatric leukemias. On the other hand, I don't know. And I think until we go and do the gene testing, we won't know. We've now seen one child who started out with ALL, typical 12-year-old ALL, and then after chemotherapy developed MDS and now has relapsed. So I would have said before, this is a myeloid problem. But, once you get a gene, suddenly it turns out that everything you thought was so simple isn't so simple. And I'm not sure I know. All that being said, people have looked in some broad populations of some MDS cases in adults, and some leukemias in adults, and not found this as a critical germline driving factor. So my guess is that the people who have germline GATA2 mutations are going to show up, most of them, earlier in life, and they'll probably have some problems in childhood, but the severe ones won't begin until they're old enough to begin to exhaust their marrow. That's my guess. Yes, sir.
Hi, two questions. The first one, apparently, the New England Journal of Medicine recently reported that the changes in Mick Jagger actually genetically induced.
You know, I didn't review that paper. Yeah.
It's not GATA2. But the other question I had, you showed a few patients with a religious phenotype.
Yes.
Is GATA2 related as well?
That's GATA25, yeah. Yeah.
The most serious question, you have in the syndrome, you have changes in B cells, macrophages, and NKs. But a lot of the papers just say deficiency in NK cells. Why is that? Did those patients really just have NK mutations, or did they have always BC, B, and NK deficiency?
I'm not 100% sure I understand your question.
When you show the patient with Christine Byron,
Yeah,
the paper just says severe BCD in a patient without NK cells.
Right.
My question is, do all the patients always lack NK, B, and BCs, or do they just have selected?
Okay. So the simple answer is nothing is always here. And the general phenotype is still that when people get sick, they have very few B cells, very few NK cells, and very few monocytes. So when they get sick, all those numbers are at about 10% of normal, 10 to 0, 10% to 0.
Really all of them.
Most people have all of them. However, that's not always true. And the variation within families is still very troublesome to me. So, for example, I have one family in which the proband got sick at about 16 and had a bone marrow transplant at 50. Her father is in his mid-70s, and his only manifestation is pharyngeal cancer, which is HPV positive, and a renal cell carcinoma. He's got a perfectly normal number of NK cells, monocytes, and B cells, and yet he's got virally mediated cancer here and another cancer down here. You could say he's got bad luck, but personally, I'm not a big believer in bad luck.
And given the big deficiency in NKs, do you ever see solid tumors?
We have seen about, out of our 25 women, 4 of them have had breast cancer. And we've had colon cancers and melanomas, no brain tumors. So those, melanoma, colon, and breast so far.
Thank you.
Ivan.
There are several transcription factors that have been shown to be monomerally allergically expressed in lymphocytes, in immune responses, T-bags, and others. Is it known if GATA2 can be expressed monomerally? Possible that some cells activate the longer immune and end up being completely GATA2 deficient in these patients?
So I think your question is there allelic activation that could be aberrant?
But some transcription factors are expressed only from one allele and not from 2 alleles at the same time.
Yeah.
And mechanisms are not completely understood. I'm wondering if this could be happening here and it could end up with cells that turn off the healthy allele and are left with the abnormal one, maybe more GATA2 deficient than the other cells.
Oh, I see. Well, that's a good question. So the question is, could you have modulation of GATA expression by shutting off the wild-type allele and leaving you with only the mutant allele? I guess that's possible, and that might explain some degree of variation in patients. The only answer I would have to that is that when we look using nanostring at specific transcript levels in B cells, so we've tried to take immortalized B cells and do all of our experiments in those so that we've got the same number of input cells in everybody. When we do that, the transcript levels are all about equal. So, I mean, the transcripts, because that's true whether we have, complete deletions or, point mutations, I think that we're expressing off of the only allele that can be expressed. But I don't know enough about allelic exclusion, and I don't know enough about any sort of transactivation. We think most of this is cis-acting, and we've got a few patients for whom we don't have mutations, but we can show that they're only expressing one allele. And so we assume that there's going to be a cis-acting element that's further away than we've completed our sequencing, but I could be wrong about that. Yes, sir.
It has been reported that nasopharyngeal carcinoma has been going up over the last years. What percentage of those carcinomas have got that indication?
Don't know, haven't looked. I think that's a very good question, and we'd be very, I should say, I would be very interested in having somebody do that work. The people in my laboratory would make sure that I never got back to Washington if I said that I would do all that work, which would mean that they would do all that work. But I mean, I think that this is the next sort of question. How can we begin to look at the spectrum of dysfunction in this gene that might be responsible for certain levels of NK dysfunction that might actually be important in solid tumors or in other types of syndromes? I think this is a very important question.
Can I ask a clinical question?
Yes.
So, in the hematology clinic, we're often faced with aplastic anemia patients, and of course the first treatment option is transplant, and the matched sibling would be the best option if it's available. When do you recommend that screening for GATA2 is done? Because unfortunately there's not a quick screening, so you'd be waiting for a genetic test to turn around, and it's also advantageous for the patient to actually go to transplant sooner rather than later.
Well, so the question is, when should you screen in an aplastic setting or in any setting?
Characteristics about a patient, because right now aplastic anemia patients are routinely screened for Fanconi's anemia, as well as
And how do you do your Fanconi screen?
with chromosomal breakage.
And how long does that take you?
A handful of days.
A handful of days?
10 days to 2 weeks.
Okay, that's 2 handfuls. Okay, I mean, just to be clear, okay, 10 days, that's 2 handfuls.
I have had it back in 48 hours once.
Yeah, but that's because they were making up the answers. Well, I don't know. I mean, I'd be surprised if you could do it in 48 hours, right? Doesn't it take, it takes DNA replication. Well, at any rate, I mean, so I would think, so this is a bigger question, really, and that, if I could restate your question, how much genetic screening ought to be done for certain diseases where overall, I mean, what's the hit rate for Fanconi's and aplastic anemia? 5%? So it's low, but that doesn't mean it's not important, and it's really important to figure that out before you go in and use a lot of chemotherapy, and to figure it out before you identify your donor. I don't know, but I would guess that the rate for this disease might even be higher, at least in the series that we've looked at so far, than for, say, Fanconi's or some of those really rare syndromes. So I think what it requires is some laboratory, and there are commercial laboratories that do this, and the genetics laboratories have to get used to the idea that people really care about genetic answers, and they can't say, well, we're backed up for 2 months. I can say that because we don't charge, but they can't say that. And so you'll have to find a laboratory or develop on campus and say, here's the screen that I have to have before I'm ready to do a transplant in this child. But I bet you're not going to transplant in less than a week for most people. So I think you could get the answer back in that time if you wanted it. We don't have a good flow assay. The level of the protein is very low, and the half-life of the protein is very short. So we need to find a new way. We're trying to figure this out in the lab. Can we use a protease inhibitor or a proteasome inhibitor or something like that to see if we can't get enough protein to be able to screen for it. But so far we don't have a really fast assay for you.
Just wanted to comment on the T cell compartment in these patients. Would you expect, since T cells can last much longer in the body than other things like populations, that if some of these patients were able to survive, they may start seeing T cell defects? Or have you looked at current patients that they're having that they lose a lot of the naive T cell compartment are very good?
Yeah, so the question is, what about T cell defects, and are they will we see more of those as they survive longer, or are they there and we're just not paying enough attention to them? It's probably the latter. I mean, some of their T cell numbers, so when we started working on this 20 years ago, what we noticed was that if we took the T cells from the patients and we mixed them with monocytes from normal donors, we could make, mitogen-activated T cell function completely normal. You took the patients, I mean, you have to have monocytes to make T cells work. The lymphocyte people don't tell you that, but you have to have the myeloid component to make the T cells do anything good. And once you put that in there, the T cell function to mitogen is normal. Now, those issues are about memory are much more complex, and we've not looked at that. I would guess that their capacity to handle all that stuff is not all that good. But, we've not seen things like thrush, we've not seen pneumocystis. I've not yet seen a case of zoster come back. So I think that most of their T cell memory functions are intact, at least up until the time that we get them to transplant. But I don't know what the future will bring for that. It's a good question. Colin.
Steve, I love how you spelled out the difference between the penetrant issue versus the variable capacity issue. And it's just really sort of a comment, I'd love your opinion on this. I'm wondering whether the next real gold mine is going to be to look at, for example, families where you've got these, there's a penetrant mutation, but some of these patients, some of them are 5, and some of them are 6. I'm wondering what the differences are there. And similarly, with the twins that you mentioned, with Miriam's story where we're dealing with XIP, we have a half brother who's got type 1 diabetes.
Right.
And I believe has EBV on board.
Yeah.
But he basically doesn't have XIP. And I'm just wondering whether that's going to be the next thing to figure out now that we get more reporting of mutations, we're starting to screen more frequently, and we're getting more coming in. Are we going to get to the point where we have a big enough N?
Yeah. Well, we're, is that the next gold mine? God, I hope so. I mean, we're investing heavily in it. I mean, the idea that variation within families ought to be genetic. The only thing I would say is that all of that hinges on the idea that it is genetic and that it's going to be exomic, right? So the couple of things that could screw you up there is that it's epigenetic, and, Mick Jagger's an example, or that it's not exomic. And I can tell you that, it's not like we didn't work very hard to find this gene for a long time. And we did every possible assay, including whole exome screening. And we missed it on whole exome. We just flat out missed it. Whereas Matthew Collin found it. Well, why? You could make all the comments about, my laboratory that you want. But the truth is that the reason we missed it is because I made the mistake of taking 2 people who happened to have intronic mutations into my whole exome screen. I didn't know that. I would have sworn they'd be exonic. And so we specifically overlooked the gene because we had 2 people who were mutated and 2 people who weren't. And he was lucky that he chose 4 people who had all exonic mutations. So I've become very humble about all the things that are going on in the majority of the DNA in the body, which is not exonic. And because we think that a lot of this is happening in cysts, and we think that there's a lot of complex regulation that's going on, I'm not so sure just where the things that affect it will be. So I think we'll go down the whole exome pathway because that's the one that's available. But if that doesn't work, I'm open to the idea that whole genome might turn out to be more important, even though I'd have no idea how to interpret that yet. Yes, ma'am.
I was wondering whether the relationship of causation in addition to the beta mutation.
I'm sorry, say it again. I didn't catch that. So the one word again, what kind of patients? Oh, why do they get?
Apart from beta.
Why, you mean why do they get those additional mutations?
Or do you see like common mutations that are common to them in these patients, and that's what's causing the difference in the presentation of the symptoms?
I don't think there's much genotype-phenotype correlation. I think most of these mutations are producing a very similar disease. I think in general in immunodeficiency, most of us have been disappointed at how little genotype-phenotype correlation there is, given at least how much time we spend searching for it. The secondary mutations that occur here it's really those trisomies. I mean, trisomy 8, monosomy 7, rearrangements on chromosome 1, and these are precisely the kinds of things that get you in trouble in other leukemias. You know, one of the things that there are some rearrangements on chromosome 3p that are relatively common that involve EVI1. EVI1 is very close to GATA2, and rearrangements that involve RPN1. RPN1's 200 kilobases from GATA2, and I think a lot of those rearrangements that are in that region are things that are going to be important in GATA2 regulation. That'd be my guess. But exactly why they happen, why does DNA, I mean, this is not thought to be a DNA cleavage or recombination factor like Artemis. So I can't explain why there's a higher rate of monosomy, trisomy, and so on. I don't understand that stuff. Sir? That should be the last, I think. Yeah, go ahead. Last.
These children get vaccinated, and some of the vaccines are animated viruses. Did you ever find out retrospectively that the side effects from the vaccination?
That's a really good question about vaccines. So, you know, in those patients from Europe, the only one who got sick with BCG was a boy in England who got BCG vaccinated at age 14, because the British are just that way. And all the children who got vaccinated at birth with BCG have been fine. Nobody got in trouble with vaccine strain of varicella, as far as I know, and nobody got in trouble with MMR and so on. I mean, you heard in the case today from Marion. I mean, nobody got in trouble like those things.
And polio is not given anymore.
Not live polio. I don't know about in Europe, but I haven't heard about that. So far, and I think in part that's because early in life they're relatively okay, and later in life they become more susceptible. All right, I got to let you go. There's a game tonight. Good luck to Michigan.